Alright, welcome to the next session of notes for biology, well, for pre-AP. Please make sure you have your learning targets out as well as the learning handout. We will go through each one of these targets as we go through the notes. So, first target we're going to hit is explain the significance of the selectively permeable membrane to the transport of molecules. So what is a selectively permeable membrane? I want you to think of if you've ever cooked pasta and you've got to drain that water out of the pasta, you've used a selectively permeable container. That container is called a colander or strainer. Okay, same idea here with the cell membrane. The cell membrane forms this really nice tight connection between the phospholipids. Okay, however, certain molecules are able to move through, in, and out of that membrane. Okay, a selectively permeable membrane, basically here it means that you've got some molecules like water that are small enough that they can fit through certain channels. And then there are other molecules, like, I don't know what this would be, but other molecules that are just too big, and so they can't pass through that membrane, at least not without some help. So, some substances can get through, some cannot. Glucose and other ions have to use transport proteins, either because they're too big or they've got some kind of charge that will not allow them to pass through that membrane. Okay, and then there are other molecules like oxygen, carbon dioxide that are small enough and don't have a charge that are able to slip through the membrane. So if you imagine with me for a moment, you're in some really populated place. Oh, I know. Think of the hallway up by the office, the beginning of school. It's crammed full of people, or even the hallways when classes get out. Okay, crammed full of people. So who's going to have an easier time getting through the hallways? Someone who's really big, or let's say like a little mouse. Okay, a little mouse is going to have a lot easier time moving through the hallway than someone who's bigger. Okay, that's a selectively permeable membrane, and it's important because the cell needs to have new things like oxygen come in for certain types of reactions that we'll talk about later. And it needs to get rid of things too. It needs to send things like proteins to other cells, and it needs to be able to get rid of some things that it doesn't want, or some things that can harm it. That's why it's important. Next learning target, compare and contrast passive and active transport. So like the name suggests, passive and active transport, one, passive does not require energy. The other, active, requires energy. Okay, now on your guys' sheet you have those two circles. I would like you in the middle to fill in what they have in common. What they have in common is right here. Okay, they both are going to be moving molecules in and out of the cell. What they do not have in common, we've already talked about. Passive does not require energy, active does. Okay, it requires something like ATP. Alright, passive, you also need to know, move stuff with the concentration gradient. So concentration gradient, if you're moving with it, you're going to be going from high to low. Okay, active is going to be doing the opposite. It's going to be going from low to high. So make sure you have that written down somewhere as well. Alright, next let's talk about diffusion. Okay, diffusion is simple blind luck, if you will. A whole bunch of molecules, remember molecules are in motion, and they're blind. Okay, well they don't even have eyes, but they're going to be bumping into each other, and when they bump into each other they're going to be flying off in different directions, and just some of them are able, well, diffusion, what we're talking about, when they're bumping into each other, they're spreading out. So if you think about when you're sitting in class, if someone sprays some perfume or some cologne, it diffuses through the air. Right? That's why you can smell it, because it's bumping around, being spread throughout the air, colliding with other molecules in the air, being bounced off those until it finally reaches your nose. Okay, so when we're talking about diffusion, there's no rhyme or reason to it. It just is a simple spreading out of molecules. Okay, So you can see some molecules in motion over to the left, or at least you could. Diffusion, when we're talking about diffusion, we're talking about molecules that move from high to low concentrations. They are trying to reach this equilibrium. Everything wants to be equal. Okay. When you talk about, rewind and talk about solutes and solvents and the solutions that are inside and outside of a cell. Okay, remember a solute is what's being dissolved. So you can have molecules or ions. The molecules can be solid, so you could have little grains of salt. You could have liquid, like water is a molecule, or some kinds of gas like carbon dioxide or oxygen. Okay, or they can be cations or anions. Cations got a positive charge, and anion has a negative charge. So an example of an anion would be like chlorine, Cl, just all by itself, or Na, sodium, after they've been dissolved, right? Those become a cation, anion. Okay, and then we talk about solvents. When we're talking about biological things, which, since we're in biology, we hopefully will talk about that quite a bit. Biological solvents are typically going to be water. Okay, diffusion is how oxygen, carbon dioxide, and other small molecules are going to pass in and out of a cell membrane. Then we need to talk about osmosis. Okay, osmosis is a type of diffusion. Okay, it is diffusion of a solvent, and since we are talking about biological things, we're talking about water usually when we're talking about osmosis. Okay, then we need to talk about this word, tonicity. Okay, hopefully you can see here, tonic, 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 tonic. So we're going to break words down. So what does tonicity mean? Okay, if something's tonic, or has, well, rewind, delete, let me try that again. Tonicity, we're talking about the ability of surrounding solutions to cause cells to gain or lose water. So when we're talking about all three, hyper, and hypo, we're talking about the movement of water. Okay, now this gets a little bit tricky because when you look at these terms, isotonic, we're talking about the concentration of solutes. Okay, so if your solutes are equal, your solvent is going to be equal as well. That concentration. I'm not talking about volume, we're talking about concentration. Okay, hypertonic is when you're going to have a higher, hyper, make that tie. Okay, hypertonic means you're going to have a higher concentration of solutes, which means you'll have a lower concentration of water. Hypotonic means you're going to have a lower concentration of solutes, which means you will have a higher concentration of water. Okay, and then this big word here, osmoregulation, that is the control of water balance. Okay, so osmoregulation becomes really important because if a cell is put into a hypotonic solution, okay, hypotonic means that its concentration of solutes is going to be lower than it is inside the cell. Okay, and if that concentration of solutes is lower inside the cell, that means, if the concentration of solutes is lower outside of the cell, okay, then the water concentration is going to be higher outside the cell because there's less solutes taking up volume. Okay, so what's going to happen is that water is going to move into the cell and move in, and the cell is going to kind of explode. Sometimes it explodes, sometimes it's able to kind of withstand it, but if it's in there too long, it's going to explode. Okay, hypertonic solution, this is when you've got a cell that has more, remember, higher, so it has higher concentration of solutes than outside the cell. Since it has a higher concentration of solutes, it has a lower concentration of water than outside the cell, and so that water is going to move outside the cell and it's going to shrink, which is why you see these spots here, because it's shrinking and shriveling. Okay, not good. Either one of those are not good. Okay, the ideal is the isotonic solution. So plasmolysis, this is when a cell shrinks. Cytolysis, this is when a cell bursts. So next we talk about facilitated diffusion. Hey, remember we talked about some molecules are just too big or they've got some kind of charge, so they're not able to pass through the phospholipid bilayer. Okay, so this is movement coming across the phospholipid bilayer, but they've got to have this protein in here. Alright, so facilitated diffusion does not require energy, which means it's going to be passive diffusion. Some hydrophilic, remember hydrophilic means they're going to love water. If they're hydrophilic, that means they've got some kind of charge, because they're going to be attracted to the charges of the water. That's why they love water. So some hydrophilic, other hydrophilic molecules can pass through them. So hydrophilic basically means we're looking at some kind of charge. So I'm just going to randomly pick here a couple. We've got some negatives, some positives that are going to be attracted to charges in here. Wow, that was a really bad plus. There we go. Negatives, positives in here. So those charges are going to attract and allow for the movement of those molecules through. Okay, other kinds of molecules or other kinds of facilitated diffusion are going to require molecules to bind to it. And this allows protein to dump molecules out on the other side of the membrane. Okay, so facilitated diffusion, another example is aquaporins. Aquaporin here, it's just a protein channel for water. Hopefully you can see aqua and you can relate that to water. Aquaporins are going to lead to rapid diffusion of water in and out of certain cells, like plant cells, like we talked about before. Red blood cells, which is why it's so important when you get an IV that they don't put in just pure water. They put in a saline solution, okay, because they don't want all the water causing your cells to, have some issues. Kidney cells, another example. Active transport, these are the kinds of proteins that are going to be bonding to some kind of molecule. So what happens here is, if you see we've got a molecule, we have more molecules outside of the cell, so we're going to say that the solution outside the cell is hypertonic and inside is going to be hypo. Okay, and so instead of going from the higher concentration down to the lower concentration, right, traveling with that concentration gradient, we're going to have this pump in here that's going to take this solute, bind it, okay, bind it into this enzyme here, and then that enzyme or that protein's going to change shape. Doesn't necessarily have to be enzyme. Protein's going to change shape and spit it out here. Okay, so that moves against the concentration gradient, which means that we are looking at active transport. Okay, it is carrier mediated. It always uses a carrier protein, and it always requires an addition of energy. Okay, so remember when we talked about breaking ATP down into ADP, we remove that protein, or not protein, sorry, we remove that phosphate. That phosphate binds here and helps that whole process. Okay, and then when it's done, that phosphate is released, and guess where it goes? Oh, it goes on to ADP. Alright, so sodium-potassium pump is really important. It's important that you know this here, that 3 sodium will leave a cell and 2 potassium enter a cell. It requires the help of ATP to power this process here, right? That phosphate getting added on there, breaking ATP down. It requires the help of that energy from ATP to change shape, and what's going to happen is it's going to take 3 sodium, kick it out of the cell, take 2 potassium, put it into the cell. Okay, 1/3 of all energy used by cells is used to drive this pump, so it's rather important for life. Okay, then we talk about endo and exocytosis. Okay, endocytosis, we are looking at a cell coming in, receptors here, swallowing, if you will, different proteins. Dang. You know what? I'm out of time, so I'm going to have to do this on a second slide. Bye. See you tomorrow.